Hi everyone, I am Saniya and in this video I'm going to be showing you how to do leftover conversion. So transforming genomic coordinates from one species, one genome build to another genome build or another species. So how do we do leftover? Using the University of California, Santa Cruz Webtool. So UCSC's Webtool. So I do have other videos explaining about genome conversions between like human genome hg38 to hg38 or vice versa, or how we can use our track layer to do leftover for a genome-wide association study, how to do the mapping of those single nucleotide polymorphisms from one human genome build to another. What are those chain files? So I do have those videos, so please do check those out. Please do subscribe. Please reach out with any and all questions that you have. But I sort of want to show you a very quick tool for doing leftover analysis on the web. So it doesn't matter how much programming experience you have. And if you don't know programming, I highly recommend it. And I do have some videos out there. And if there's anything you want to see, please do let me know. But no matter what, if you have a chromosome number, a start of a region and an end of a region, and you know what build it is, how do you just transform it from one to the other? So in this example, I just have these 28 genomic coordinates on these two chromosomes for this toy baby example, chromosomes 1 and 10. And I want to convert them from this rat genome. So it's taken from a rat. And I want to kind of do this leftover to map those same coordinates, not just in a human hg19, but in an hg38. And the way to do that wasn't exactly the most intuitive on the tool, which is why I want to show you that there are two steps to doing this process if you want to go to hg38. But I just kind of want to walk you through this example. So let's get started. And there's so many different tools out there to do leftover analysis. So if you find that our track layer in R is maybe breaking apart intervals into smaller pieces, and you want to have longer, more contiguous pieces, when you're mapping from one genome to another, maybe you then want to check out this tool here. And there are a lot of different parameters that you can experiment with as needed. So here in this example, what you're going to see is that we have these rat coordinates here. So it can be a bed file or it can just be anything. And as long as you can open it, and you're going to see that you have a chromosome number, a start of a region, and an end of a region. So for instance, the width of this region over here is taken by this end minus the start here. And we can see that each of these has, around between 149 and like 300 or so base pairs. So this is 149, it's this chromosome 1 region. And it's in a rat. It's rat and it's an rn5 version. And there are multiple different versions because, biology is only getting better and the resources and the tools that we have are only improving. So what you do here is you see this chromosome number and the start and end position. So we can just see, okay, well, what are these in humans exactly, right? Because there's so many experiments. And I know that we have people for the ethical treatment of animals, this PETA out there, and there's a lot of rights for animals, and I'm a huge supporter of animal rights as well. But there's so many experiments out there that are conducted on these animals because it's safer or they have a shorter lifespan and they breed faster so you can observe results in generations, or for whatever reason. We do sometimes these scientific experiments where we can try to knock out a gene or do whatever it is, and then try to figure out, okay, well, this happened in a rat. Well, what is this going to be in a human? Or maybe we do this in a cat, or we do this in a fruit fly, or C. elegans, the worm, or we do this in one of our closely related species in a monkey. We could be doing this in other species, and then we want to figure out, okay, well, what is this when it maps from a rat to a human? What are the coordinates? Okay, we did it here. It's like we wrote this book and it's in Hindi. But what is it? This beautiful book or this beautiful movie. How can we make it accessible to people who speak English? And I know I can try मैं कोशिश करती हूँ to talk in Hindi, but I can't really read like the Hindi scripts. I'm not really skilled, so you would definitely need to translate that for me. The only thing I know is probably Om. I have like this necklace and it has like this Om here. So anyways, that's just besides the point. That's just a little tangent. But what I'm trying to say is that it's really important to be able to do this conversion. And a lot of the times we have these studies and we want to figure out what does it look like in a human at the end of the day. And just to kind of make it a little bit lighthearted, because that's sort of my signature style, I just sort of want to show you that we just want to go from a rat to a human. At the end of the day, that's all we really want, rat to a human. There's just so many things, like a little rat sitting here. That's all we want, these poor guys. Alas. Okay, so we have these coordinates. So what do we want to do? So there's this tool here, and I'm going to put this in the bio as well. So do check this out. This is the UCSC Liftover tool. And remember, they are the ones who have these chain files that we can download, and we use them in my other video as well. So this is genome.ucsc.edu/cdi/bin/hg liftover. So this is liftover analysis right here. And this is what we want to do, is we want to convert, again, what our goal is we want to convert these 28 coordinates that have some relevance to the rat. Maybe the regions of open chromatin, or maybe some gene is it, these are different gene regions that are being expressed, whatever it is. These are just some made up coordinates here, and there are 28 of them. So typically in these bed files, you will have a bunch of different columns. Like after the third column, there are like some different annotations that can be there. But the first three are really crucial. You need the chromosome number, CHR. This means chromosome 1. This means chromosome 10. And the importance of this is, like, we have different genomes than these rats, fundamentally. Like, we have this rat genome right here, and you can see that this rat has like a different this is a Rattus norvegicus here, and we can see that it looks, this karyotype, this breakdown of its chromosomes is different than what we see in humans. So here we have like 20 of these chromosomes in rat, then we have this X and Y here. So it's kind of different. Like what we see for humans would be the following. If we had to list them out, I know like this looks kind of doubled up from each pair and whatnot, but like you can see here that we have these 22 pairs of autosomal chromosomes, and then the X and Y for males and the XX for girls. So the genomes are fundamentally different as well, but there are some similarities. That's why we do those studies ultimately. So Liftover is a great way to make sense of that. So we're going to go back to this tool. And I hope that this can motivate you as to why this is important to do Liftover. It's because we're trying to map these regions that we have identified in rats or whatever species it is to humans. And what you'll notice about this Liftover tool as of now, as of the time when I'm making this video, is that it currently is not super accessible to map them to hg38. So use a bit of a workaround, but I just want to show you so you could be creative when you're looking at this as well. So we can see that there are lots of species, and this tool here, this tool And this is like the same site that has those chain files. So this tool here does lift genome annotation. So it converts genome coordinates and annotation files between assemblies. The input data can be entered into the text box or uploaded as a file. You can upload it as a file. We're going to enter it into the text box in this example. So for files over 500 MB, you can use the command line tool and this liftover documentation. If a pair of assemblies cannot be selected from these pull-down menus, you might need to do a sequential lift, like MM9 to MM10 to MM39. So that means is that you might have to do this in processes. So we want to ultimately go here from this rat genome all the way specifically to the most modern currently human genome reference build, like one of the more modern ones of hg38. But that's not going to be possible right away. So we're going to do this in 2 steps. And the 2 steps that we see here, the first step is going to be rn5 to hg19. Then the second step will be converting this hg19 file to hg38. So it's going to be these 2 steps, and that's what we're going to do. So we can go here and we can see there's so many things. There's even a turkey, you know, in the spirit of close to Thanksgiving time. Panda, they have an opossum, Nile tilapia, a green monkey, gorilla, golden snub-nosed monkey, so many different dogs. Oh, wow. Cow, so many different things. Human. And what we see over here is that, like, for the rat, let's see. For the rat, we have a rat here, and we have so many builds. We have this November 2020 rn7, which is the latest one as of now. Then there's one in July 2014, rn6. Then there's one in March 2012, which is rn5. So we have that kind of data. And it really depends if you're working with different collaborators or whatnot, or your rest of your data is in a certain genome coordinate system. Sometimes it's just better to stick with those defaults. So you'd wonder, okay, well, why not like just push her to the most modern ones? Well, sometimes, you know, and I work with this as well, is you could be working with very old data. So some of the data I've looked at has even been from the microarray, and I've had to map those probes to genes and so on. So some of the data can be just like really old, and that could really be like what you have to work with. So what would be better then is to figure out, okay, well, how can I make the most of the rest of the data in the experiment? And that's what you'd want to stick with. So in that case, like what we see here is that I have this rat rn5. And now if I look in the human, so they say, okay, for rats, this is your original genome. This is the assembly. So I'm saying that this bed file, if you will, this data that we're looking at, chromosome number starting, and this is some rat genome rn5. So it's a relatively older build. And I want to then convert this to the human genome. So then for the human, you know, so they think from rat, we can convert to human, mouse, chimp, rat, rhesus. So for the, for the rat, what we see is that we have like rn6 or rn4. They still don't have the rn7 conversion. So for the human, I want to convert it to hg38. So will that be possible? Let's see. And you probably, if you've been paying attention, you would know that that's not going to be possible as of now. But if the tool changes, don't blame me. I mean, it'll be better for you guys if they have the hg38. But I just want to show you that this is a very accessible tool for you guys. So we are sort of limited if we use this tool by what they have available. But there are so many other tools out there. If you want to use R, if you want to watch the other video that I have, you can see that and try that out. So for the rat to the human, this is the mapping that we're looking at here. This is the rat to the human. Now let's see if we can find hg38. No, we don't have that. We have hg19. So we have to think about it. It's kind of like a puzzle. Okay, they don't have hg38, but once I convert this rn5 to hg19, what can I do next? Then I can say, okay, if I have human, which is at the top, If I have the human hg19, which is from Feb 2009, grch37, and they have these newer builds as well too, but we're just sticking with hg38. But okay, hg19, then let me see if I can convert hg19. If I can convert this to, yep, I can convert this to hg38. So solid. So the strategy will be that I'm going to look at going from the rat over here, and it just takes a while to reload it. I will go from rn5 to hg19 in the first conversion. And then in the second conversion, I'm going to take those hg19 coordinates, and then I'm going to map them to hg38. And even between these builds and assemblies, as you can see, even for the same species, technologies improve as well. Like we go from this microarray data for gene expression to RNA-seq levels, that could be in bulk in tissues. And now people are going inside and collecting the single cell data as well. And the data is always getting better. And whatever we had before is iterative. Things are improving and becoming more and more accessible. So even within the same species, there are different assemblies, and those could be very different mappings. And I know it is a bit tricky to put your mind around it. And I have another video just talking about hg19 and hg38. Okay, so what we see now is that this is a rat, and it just needs to be like refreshed. Okay. Okay, so we're going to look at this rat here. And for the rat, we are going to look at rn5, and then we're going to go to the human hg19. So what we'll do is now we have to put it in like chromosome number, start, and end region. That's one of the ways you can put it, or you can also put it with the chromosome number, start to end position. And if you're working with Excel for that reason, there are ways to put it into that format as well. What you can do is, it's a little trick I like to use called concatenate. It's also a programming term as well, where concatenate like lumps things together. So if I concatenate this thing with like some exclamation points and a smiley, then it's just going to concatenate that, like chromosome 1, and then a bunch of exclamation points and a smiley. So that's what concatenate does. It takes the values and it can attach other things to it. So if I wanted to kind of mimic what they could also accept chromosome number, a colon. So what I would do is I take a chromosome number, then I would also just put a colon here. And just to show you, this gives us chromosome 1 colon, what they're looking at. But then I also want to take the starting region. And you see how it looks kind of like what they're looking at? So I'm concatenating A2, this colon, this value over here. And then I want to add in a little dash, and then this end region over here. So everything is kind of separated by this comma. And if you have to add in special text, you just put it in quotes. So then this is what it's going to look like over here. So it looks just like this. So the other thing I like to do is, so you see like this is the concatenate, and then you can copy it and then paste the values, because these were like from before, this is not really values. These are just like formulas. So if I were to just copy this, it would be a bunch of formulas. So you want to really just, like if you're done with this, you want to do copy and then do this paste values. So then each of them is exactly the value. So now if I copy this, I will get like the proper one. So, okay, so you can do it either way. I'm just going to take this ABC part, and the first part you do not need the header at all. And often bed files do not have the header. They can have some other annotation, but it's kind of assumed that it's a chromosome number, then the start, then the end, and then there are like additional columns out there that you can look at. So whatever format it is, the data. So what you can do is you can see that I'm just going to paste in these 28 rows here. So you can see like at the bottom, this is chromosome 10, 387, 1432 to 387, 1581. So that's what it looks like over here. And now I'm going to do submit. So then we're going to get the results. And again, here they're like bed 4 to bed 6 options, allow multiple output regions, minimum hit size, minimum chain size, minimum ratio of bases that must remap. So there are all sorts of parameters that you can play around with. I just recommend if you don't know too much, just stick with the defaults. You can't go wrong there. So what the results will say is that it successfully converted 12 records and the conversion failed on 16 records. So if we do 12 plus 16, that is 28. And we had these 28 rows. So on 12 of them, I mean, it's not a majority mapping. I mean, a majority, like half would have been 14 and 14 failed, but it is what it is. So for the failure files, the failure messages, deleted a new sequence intersects no chains. It insufficiently intersects one chain so there are all sorts of problems. And for the failures, we can see just why these 16 did not map. So it could be deleted in new. The sequence intersects no chain. So the ones that were deleted in new, it seems like all of them were deleted in new. So they intersect no chains. And the chain file that it was fundamentally looking at was rn5 to hg19, hg19 chain file, converting from a rat to a human. So then what you can do is you can just do view conversions, and it's going to download, and you can see at the bottom right in this area here, it's going to kind of show the name of the file. So it's downloading over here, this bed file. And when you download it, there's like a lot of different tools out there. And if you have a Windows, or if you're able to get this tool, it's free. It's Notepad++. So you just can go on the web and download it. And a major shout out to my brother. He's the one who told me about this really cool tool. Thanks. Thanks, baby bro. I love you. Okay. I hope this won't make him cringe. Anyways. So yeah, he told me about this really cool tool, Notepad++, and I use it. You can open up a lot of different work with a lot of different programming languages and open all sorts of things. And as you can see, I've been visiting it very frequently, and you can just download it as needed, and it's free. So that's what you do it. So I can just like open up this file in Notepad++, and so here I've opened up that file that we downloaded. I've opened it up in Notepad++. So what we're going to see again is that this is the start. Chromosome numbers, start, and end. And what we're going to see is, okay, how do we interpret this? Well, what this will do is it's going to return output in the following format. Okay, so this was the example of that bed file here that we have. So we can just copy this over here, like to the new thing, and we're going to figure out, okay, what on earth is going on here? What did we just get? So this was the before. So this is the original file, and let's just see what this is. This is the result, and this was just 12 of them that were mapped. The other 16 just couldn't be mapped, and It's sad, but it is what it is. Like humans and rats are so different that we do not expect complete leftover as well. So this is the original rn5 here that we had start, and now we're going to just show what we got from this leftover analysis, which is here. Remember, only just 12 of them were converted. 12. So what are all these files in the output that we got? Because this is what we got when we downloaded it. Again, this is what we downloaded, hg genome, this is what we got. So what is each column? So this is the chromosome, this is this. Okay, so CHR1, 2518, 9186 to this. So what I noticed about leftover is that it may not be completely exact. Oh, my English is going for a toss. Okay. So it might not be like completely exact or accurate, right? You know, so I kind of figured this out and I was like, little stumped. Like, what is what? Like, is this, is like, what's what exactly? Like, is this the original data that we put in? Or is this the original data that we put in? Because we were doing this rn5. Essentially, we were doing rn5 to hg19. hg19. So like, what was what? I was thinking, okay, well, what is it? I was trying looking it up, and this is in a bit more like a bigger data file. I was like, what is what exactly? Then I was like, hey, so it looks like if you look at this over here, this is so this is 251. So it seems like this is the same as this, except if you look at it, and this is where like you have to see that this is like chromosome 1. Okay, that's fine. And then what really got me is that 258139484. That's the ending. And then that's also the same ending over here. And then this chromosome number is also the same. So the only thing is that this number and this number, this is original one, and then one in the UCSC results file is just one base more. The 258139185 to 258139186. This is just one more than this over here. That's the difference. And then we can just see the other results too. Like if we're looking at this, 259004266. Well, what does this map to? This seems like it has, okay, 25900. So I'm scanning across for double zeros. Okay, this one. And it seems like the ending is 4514. So they seem like they're exactly the same. So what this leads me to believe is that this is the rn5 coordinates. And I know that like this part here, this base right here, it seems like it's just one more than this base over here. And that just might be something funky about UCSCs. So don't worry too much about that. I think the most important thing is to just realize that these are the rn5 coordinates and that this is the hg19 chromosome. This is the and what you can see here is what's interesting is that it's essentially telling you hg19 start and then hg19 end. So it's saying that, hey, okay, the rat chromosome 1, these regions right here, they are analogous. I mean, if you think of it from an evolution point as well, if you look at this phylogenetic tree, if you even look at the phylogenetic tree of evolution as well, of how evolution was done. Fruit fly, human, chimpanzee, like in terms of different species over here. So this is where rat is. Rat isn't as close to the human, but human is closest to chimp, and then rhesus monkey, then bush baby, and then tree shrew. And the smaller this is, and this is like a dendrogram that we use a lot in clustering and similarity and all sorts of machine learning approaches. You know, it's kind of the same. It's like how neural networks are based on models of the brain, and that's also kind of what is an artificial intelligence driving it. So it's kind of like interconnected, guys. We don't keep reinventing the wheel, okay? Anyways, so you can see that the smaller that this is, that means that they're more similar to each other. So this means that this is almost like a group over here in and of itself. This is a group right here. And then this group is the closest because they have this edge with this group over here. So we're not the most similar with rats, but we do share quite a bit more than with the zebrafish. The zebrafish from the beginning of time and evolution. So we have, you know, it's from, this is the distance to be able to connect to fugu or a zebrafish. We have to like go up here and here and here and here and here and here, and like kind of almost go back to the beginning of the time and then go down there. So we're like as different from them as possible. But there are advantages to using each of these different species for experiments. You know, if we do ethical, you know, IACUC as well, we get the IRB approvals, and I believe we should treat the animals ethically, then use them as needed for the advancement of science. Okay, so we're not quite as similar to the rat, but still a lot closer than we would be to a zebrafish, for instance. So what it's saying is that in a rat, this chromosome region here, the CR1, chromosome 1, it maps to chromosome 10 in a human into this region right here. And we could see that this was like about 299 base pairs in a rat, and in a human, what is the base pair? We can just do this minus this, and then we'll see it's around 290. So this 299 in a rat, around 290 bases in the human. And we can again see that, okay, this is chromosome 10. I like to just be really exhaustive with the example so that you can see this is 4175, and then this one maps to this. Again, they just seem to be off by, you know, this is 26 and then this is 27, but it's like the same thing. And this is the rn5 coordinates, and then this is the hg19 coordinates. And just for the sake of it, I will say that this is the rn5 and this end is rn5 as well. So the next thing I want to show you is that we are not yet done. This is the result of liftover from hg19 to why am I so, okay, I'm jumping ahead. rn5 to hg19. Okay, so that's what this is over here. Okay, so then the thing I want to show you is that, okay, so if we wanted to just use hg19, then that's fine, and then we're done, okay? Just somehow get over the fact that 16 sequences, 16 of those little regions, they did not map. That's sad. But then if we want to do some additional mapping, and the chain files are not available on UCSC on the tool, but they're available on the web there. But again, with the web tool, there are limitations, and that's why I keep stressing programming for you guys out there. But an advantage I found from this tool is that it does have like longer sequences, and then our track layer could tend to split them into smaller sequences. So, but my video goes over our track layer, and I've also linked that in this video here. So if we're wanting to just do hg19, we're done. But if we want to do hg38, then we just have to do the same step again, but we are instead going to just take these regions right here. So before we just took like what we had put in to get hg19 is we'd put this in. Now from rn5 to hg19, we're going to just take this over here. So I'm going to go back to the web tool, and just for the just for the ease of comparison, okay, I'll just clear this here, and I'm going to go to human. And I want to go from hg19 all the way to hg38. So human to human. That's what I want. Human to human. hg19 to hg38. So what I'll do now is I'm going to take this and copy it. And then just paste them here. So just paste these 12 sequences, and then I'm going to do submit. And then it successfully converted these 12 records that we put in. Again, these 12 records were these records here. It mainly mapped these results from these chromosomes to chromosomes 10 and 16. It seems like chromosome 1 in the rat maps to like chromosome 10 in our data, and then chromosome 10 in the rat maps to our chromosome 16. But I wouldn't make those conclusions just yet. These were just very few that were mapped. But anyways, I had taken these and I got my records, so none failed because we're just mapping between two human coordinates. So of course we expect a lot of these to convert, not like we had like 12 out of 28 converting before. So these are other things here, and I can view the conversions. And again, it's going to download this other bed file, and I can open that bed file up. And a tool like Notepad++. So here, this is the hg38 coordinate. So we're going to now just put this in here. Okay, getting a bit ahead of myself here. Okay, so this is the hg19 to hg38. And I want to do my job and show you that this is hg38 chromosome, hg38 start, and hg38 end. I want to show you that it's the same, and we do expect like that the chromosomes in the human should be the same, given that we're just converting from one build to another, the same species. So we see that this is okay whenever it's chromosome 10 here. So whenever it's chromosome 10, then it's also chromosome 10 here for the human. hg19 being like that is the same here as hg38. It's the same species. Same species. And then what we can also see here is that this would be hg19 coordinates, and what we will see is, okay, this guy over here, this chromosome 10, this region right here, right? This right here, this little green guy maps right to this one here. And again, like you can see that this is 5-2, but this is like 5-3 here. But this is like 4-4-2 and this is 4-4-2. So it seems like the starting is always just like +1 when it returns it, but the ending stays the same. So it typically in this, it's going to always return like CHR and then start +1 and then to the end. That's what it seems like it's doing when it's returning the result. It's returning the chromosome and number, then it's giving like the start +1 till the end stays the same. And then over here, this part right here that we were looking at, this maps to this, and so on, the same chromosome. So this is the hg38 coordinate system. So then you can kind of trace it back a bit and then see that, okay, and similarly, I think it's going to be the case where these tend to line up. These, like all of these right here, these regions tend to be the same over here. I think it just maps perfectly. There's no like jumbling around. Whereas over here for the rat and this, like it's not like exactly. I think what it does is it returns them, but but some of these were missing and not found, and that's why you see like this green comes before this yellow, and then this like bluish guy here. And I think it's keeping that order here, but some of them were not mapped. Whereas since all of these were mapped, then this maps to this maps to this to this, this to this, and so this to this, and so on. Oh wait, my bad. Oh wait, I meant this to this, this to this to this to this, because these are the rat coordinates. But like if the chromosome 10, this is the, so that's what I wanted you to see. And ultimately, now that we have this, we can just say that, hey, altogether, what I can tell you is that from these examples is that like if we have this little guy here, this is rn5, hg19, and then hg38 for this region. So let's just look at these three examples. So let's just take this guy, and this guy. Then the hg19 coordinates will be the following. hg19, the hg38 coordinates I would get by just doing concatenate on this. I should do concatenate for this. Oh gosh. Start to the end. So this is the hg38 coordinates. And I'm going to take this. I'll just do copy and then this paste special. And then I'm going to take these right here. And then I'm also going to go across and take him. Okay, so this is the result of the sequential liftover. So we're going from this rat to this human. Sorry, there's a little rat. I know Stu a little as a mouse, but rat to human. Okay, so in a way, the rat, we first mapped it like sequentially as follows. So just to kind of wrap up, what we did, we used a tool that we went from rn5 to hg19. And you can see that these are the coordinate systems, chromosome 1, and in these three examples, out of the 28 records, 12 were mapped to the HG human reference. So this is a rat genome assembly, 5 to the human genome, hg19 to the human genome, hg38, which is the newest one. Oh, no, not the newest one, but a newer one. So we mapped these to each other. And then what we did is we saw how these chromosome 1 of this rat maps to this hg19 chromosome 10. And then for each of these, we kind of mapped them then to hg38 here. And we can ultimately just see that what we went for is we went from this region to this region. So chromosome 1, 258, 139, 185. I also do like to have these commas. Maybe I'm a bit old school, but I do like that here and this right here. And I just kind of want to put the commas in. So it's bothering me a little bit, and maybe it bothers you guys too. Or maybe I'm just fussy. And then what you see here is that, so this chromosome 1, 258, 258,139,185, the 258,139,484 maps to this chromosome 10, 50,576,392 to 50,576,682. So this seems to be like a little shy of 300 base pairs. So that's what this mapping does. So we can kind of convert this in a rat to this in a human of the build that we want. And this to this and this to this. And we just had to go through this by this intermediate. So that's sort of the way that I recommend looking at it. And I hope that this was helpful for you guys and that you can use this tool to kind of do this mapping. So we did these 2 sequential mappings using the UCSC tool out there that is free. So we had done, again, we had, just as a quick little recap, we had gone on this tool and we had gone from the VAT rn5 to hg38. We had put in our 28 sequences, the regions, the chromosome number, start and end. And then we had just clicked submit. We got the conversions, we downloaded those. And then the first 3 columns are the new coordinate system that you wanted. And the column to the right, which has the chromosome numbers and everything else appended like that, like this would be the old. This column is the old. This is the new chromosome number, new start, new end. And this is the what you just gave it in, the old. And similarly, like for the VAT, this was the VAT coordinates. This was the human start, hg19 start, hg19 chromosome, hg19 start, hg19 end. And these were the VAT. So this was the old. And then this part right here is the new. The new was all of that here. And then that old was that. So that's what that was over here. So we had just done, we'd taken those 12 and then we kind of put in the 12 here when we were doing hg19 to hg38. And you can do this as needed. And as I said, you can keep doing some sequential conversions. So if you want to do MM9 to MM39, you might have to do MM9 to MM10 and then MM10 to MM39 as needed. So I hope that this is helpful. Again, please do try it out. Please let me know if you have any questions. Please reach out as well. And please don't forget to like, subscribe, and share. So I'm Saniya and I'm really happy to be able to help you guys with Liftover. And I do have other tools and resources out there as well. So thank you guys and have a wonderful day.